Well, thank you, Chen, for your nice introduction. So I also want to thank Mike for organizing the exceptional conference and most enjoy so far. I'm delighted to have this opportunity to present our work on discovery and the initial characterization of a new protein modification pathway called lysine lactation. My lab is interested in refining proteomic technology and using this technology in combination with other tools to discover new cellular pathways with a current focus on PTM pathways and epigenetic pathways. We also use these tools to study functions of metabolites. We hope this study can lead to a better understanding of diseases and eventually to new drug target and therapeutics. So as you know, there are four major types of molecules: DNA, RNA, protein, small molecule, or you can, now you talk about metabolite, theoretically we can also argue about carbohydrate, right, glycolipid. So if we look at literature, so there are so many tools available for the studies of protein, DNA, and RNAs. And therefore, we have much more understanding of these three groups of molecules. In a big contrast, we know much less about functions of metabolites. Whenever we talk about metabolites, we usually talk about their metabolic function or as an energy source, but their signaling function or regulatory function, we know very little. So there are about 110,000 small molecules in mammalian cells. Can you name how much we understand their signaling function or regulatory function? I think it's reasonable to say that maybe less than.
Where is that?
Oh, from the review. From the review. Not a single mammalian cell, but together in mammalian cells. Yes. So I can send you a paper if you want interest. So it's fair to say maybe it's likely less than 100, right? So personally, I believe non-metabolic function of small metabolites represent dark matter. So we truly know very little about their signaling function. In this slide, I list 7 categories of major issues we want to understand in order to characterize metabolite functions. Because of a tremendous effort from the chemistry community, we largely have an encyclopedia of all the metabolites or small molecule from mammalian cells. Because of quantitative metabolomics, like what Mike just talked about, we also have significant understanding of their dynamics among different cellular conditions. A big gap is metabolites and protein interactions. It's very difficult to do. And metabolites can inhibit or activate protein's function. Metabolite can mediate signaling events. How much we know? We know very little. And another issue is that metabolite can be precursor for protein modifications. A typical example is acetyl-CoA. Acetate is a precursor for acetyl-CoA, which in turn serves as a cofactor for histone acylation. These information are critical to the functions of metabolites, and this information can be invaluable for new drug discovery. Related with this issue, I can share some examples. As you know, there are oncometabolites. If you look at history of anti-cancer drug development, so all the target is based on expression, mutation, or candidate-based molecular mechanisms. A major difference between normal and cancer is metabolism. So significant number of metabolites are upregulated in tumor cells. Did we take advantage of those information to identify novel drug target? Yes or no? Even for those ones like IDH and ketoglutarate, right? You can argue this still from mutation analysis, originally at least, right? So therefore, we still have a big gold mine we haven't yet fully explored. So also, as you know, an emerging approach to discover new drug target is like a molecular glue or protein neighborhood, right? So small molecule represent a key molecule, not only for molecular glue, but also for protein neighborhood. And do we have mature technology for those? I don't think so. It's a pain S to identify molecular glue, right? So for all these, I think non-covalent interaction will represent a key information to address these two issues. So today, so I want to share with you one aspect. We work on these two issues, metabolites as a precursor for protein modifications. So as you know, acetate is a precursor for acetyl-CoA that can serve as a cofactor for histone lysine acylation. Lactate. If you look at the structure, it's not a huge difference. This is lactate, I'm sorry. So this is lactate, right? So based on chemistry, you can argue lactate can also serve a precursor for lacto-CoA. Lacto-CoA can be possibly cofactor for histone lysine lactation. So based on these two chemical reactions, I think this is a good hypothesis. And therefore, we hypothesize that histone lysine lactation exists in the cell. Lactate is a very interesting molecule because it's associated with hypoxia, a physiological event which is associated with many, many diseases and physiological change. Also, in cancer, diverse cancer, at least solid tumor, almost all the solid tumor has a much high concentration of lactate. The concentration of lactate can be elevated from sub-millimole to 20 to 50 millimole. That's a huge, huge increase. Biological studies of upstream signaling of hypoxia led to a Nobel Prize several years ago. However, downstream event of hypoxia, we don't know. Signaling function of lactate in both hypoxia and Warburg event, we don't know. So therefore, we want to set out to check out if lactate can be precursor for histone lactation. I think this study will be also important to study tumor microenvironment because lactate is a key molecule which is highly, highly stimulated in tumor microenvironment. So we, as a proteomic person, we use mass spectrometry. We analyze histone peptides, and we ask if there's any peptide with a mass shift caused by that of lactation. So in addition to this candidate approach, we also develop an algorithm which enable us to filter out all the mass shift caused by any protein modifications, as long as the modified peptide has a good fragmentation pattern. So this is just one example from this tandem mass spectrum. We identify this H3 peptide has a mass shift of 72 Dalton over here. So these 72 Dalton based on element composition, we can quickly deduce element composition. From this element composition, it looks like it's most logic structure is lactate. So at least lactate is the major candidate for the modification. We detect more than one peptide. Initially, it's 5 peptides. So then we validate if lactation truly exists in the cell. So to do this, we make synthetic peptide. Then we compare the HPLC co-elution and the fragmentation pattern between the cell-derived peptide and the synthetic peptide to see if they match. That's how chemists used to confirm the same structure, right? Indeed, that's the case. From fragmentation pattern, from HPLC co-elution, and basically it fit histone L lactation. So at this stage, we feel reasonably comfortable. So then we develop antibodies, sequence-specific antibodies or sequence-independent antibody to validate these modifications. We also use isotopic labeling to demonstrate if isotopic lactate can label histone lactation. All these 4 independent experiments fit, leading to a solid conclusion that histone L lactation exists in the cell. So this is one example. We develop antibody against the L lactation. As you can tell, the antibody is very specific. We can carry out Western blot analysis. We can do immunostaining with the antibody. And we show that histone lactation is stimulated in response to elevated concentration of L lactate in a dose-dependent manner. Clearly suggests that, number 1, histone L lactation exists in the cell. Number 2, L lactate is the precursor for histone modification. So we published a paper about 5 years ago on the initial discovery of histone lactation and initial characterization of this modification. So this slide is a brief summary of this paper. So under diverse cellular conditions like hypoxia, tumor microenvironment, or LPS challenge to macrophage cell, or stem cell differentiation, under these conditions, depending on the context and the cell type, cellular glycolysis is stimulated, lead to high level of lactate. And this lactate, we hypothesize, is precursor for lacto-CoA for histone lactation. So increased histone lactation regulate gene expression, includes those in macrophage. We show that this pathway is mechanistically different from the popular, widely studied histone lactation. We also show that the ex vivo data we observe in the culture cell can be recaptured from the primary cell in the mice. So all our results point to a conclusion that the structure we identify for histone lactation is in L form, because LDHA, which generates L lactate, is important for histone L lactation. When we treat the cell with L lactate, histone L lactation is increased. And also under hypoxia glycolysis conditions, which all generate L lactate, stimulating histone L lactation. But in the literature, in addition to L lactate, there's also D lactate. Also, there's a small molecule called methylglycerol. Okay, these two L lactate or some other small molecule like LGSH can plus L lactate can eventually lead to delactation or KCE. These two modifications, which has the same molecular weight or mass shift, but a different structure conformation. So after we published this paper, then concern was raised. Here are a few concerns. So in addition to L-lactation, does D-lactation exist? Does KCE exist? Is L-lactate the only cofactor for histone lactation? And is this a chemical catalyzed reaction or enzyme catalyzed reactions? So we address these issues. So to address these issues, we develop antibodies. Antibody against L-lactation, against D-lactation, and against KCE. So their molecular weight is the same, but their structure is very different, right? So it's not difficult to develop antibody. So we develop pan antibody against this modification, and these three antibodies are very specific to their corresponding modifications. So then we use this antibody to examine their dynamics, if we can do IP mass spec to pull down their modification site. So as you can tell, only L-lactation responds to glucose concentration or glycolysis. So this is just one example. There are other examples too. So this is another example in response to hypoxia. Only L-lactate, only L-lactation respond. And L-lactation is also depending on concentration for L-lactate. So in addition to this experiment, we also developed chemical derivatization in combination with HPLC to determine the concentration of L-lactate versus D-lactate. We also try to check out if we can identify any KCE peptides. So at least under our system, in the cells of our interest, and we analyze. So we show that histone L-lactation is a dominant isoform among the three structure isoforms. So histone L-lactation is the isoform of lactation that is elevated in response to hypoxia, high glucose concentration, oncogenic signal, and LDHA activities. So we almost cannot detect D-lactation and KCE under our conditions by HPLC mass spec analysis or even with IP mass spec analysis. So we believe D-lactation is not significant under most conditions because D-lactate concentration is much lower than L-lactate. So D-lactate concentration is about 1,000 times less than L-lactate. So after we confirming this modification, we try to identify regulatory element for this pathway. We want to study biochemistry, we want to study their functions. So here just one example, some examples. So we show that P300 and MOF1 two acetyltransferases can catalyze histone lactation reactions. We show that HDAC1 and 3 can serve as histone delactylase. They can remove histone lactation, not only for acylation, but also for lactation. So in the cultured cells, among all the experimental conditions we studied so far, oncogenic signal can elevate glycolysis and histone L-lactation. So the next question will be how lacto-CoA is generated. Indeed, lacto-CoA wasn't reported in the literature until we report histone lactation. So, so this is an important gap for this pathway. So we hypothesize that this enzyme, lacto-CoA synthetase, which can catalyze the synthesis of lacto-CoA from lactate. So we have a bunch of reasons for this hypothesis. Number one, we can detect lacto-CoA by mass spec. P300 MOF can use lacto-CoA as a cofactor for histone lactation in vitro. And L-lactate can stimulate histone L-lactation. So therefore, the hypothesis is solid. So then we set out to screen for the enzyme for lacto-CoA synthesis. So what we did is we go to literature, protein database, we pull out all most relevant short chain CoA synthetase. Then we knock down each of these to ask which enzyme can reduce that CoA, which enzyme can reduce histone lactation. And this is what the result we observed when we knock down SEOCLG1 or succinate CoA synthetase G1 subunit. Histone lactation is drastically reduced. So what is protein? G1 protein, SCS-G1 protein is a common factor for ATP-SCS or succinate CoA synthetase or GTP succinate CoA synthetase. So these proteins were originally classified as a mitochondrial proteins. They're mitochondrial proteins. How can they catalyze synthesis of nuclear lacto-CoA and for histone lactation? So step one, we want to validate enzymatic activity. So we use the cultured cell. We did knockdown experiment, knockout experiment, and then mutagenesis experiment, expression of mutant enzyme or wild type enzyme or mutant enzyme. So we also measured the lacto-CoA concentration in response to these knockdown mutations. So clear, it's very clear. This enzyme is a GTP-SCS or both G1 and G2 subunit are critically important for the generation of lacto-CoA and histone lactation. So we also carry out enzymatic analysis, kinetic analysis. We also saw the structure of these proteins. And again, all these data confirm the enzymatic activity of this interesting enzyme. So next question will be, because histone lactation happens in nucleus. So if this enzyme is located in nucleus, so we did immunofluorescence. So wild type cell and the mutant cells. So we demonstrate that. So this enzyme can be located in nucleus in addition to mitochondria. When we mutate nuclear localization signal, so nuclear localization is largely disappear. Nuclear localization signal not only block the localization of this enzyme to nucleus, but also dramatically decrease the concentration of lacto-CoA and histone lactation. So all these data fit. Nuclear localization is critical for the generation of lacto-CoA and for the histone lactation. So we want to study how this enzyme is regulated in nucleus. So what we did is we purify this enzyme's complex. We ask which enzyme is associated with, which proteins are associated with this enzyme. So we show that when we express the flag tag G2 subunit of GTP-SCS, we can pull down P300. P300 is a major protein in this pull down experiment. So GTP-SCS can generate lacto-CoA, right? P300 can catalyze histone lactation. So this in situ generated lacto-CoA can be directly used by P300 for histone lactation, right? So this is what we observe. We observe together. So I don't have time to go to very details. So all our biochemistry, structural biology, metabolomics, proteomics, and the mutagenesis experiment and enzymology suggest GTP-SCS is a robust enzyme for the generation of lacto-CoA. And this enzyme can associate with P300 and to catalyze histone lactation. So this is the model we have. So under diverse conditions, when the glycolysis is elevated, leading to high concentration of lactate. And this lactate can be used by GTP-SCS to generate lacto-CoA. This lacto-CoA can be used in situ for histone lactation. The upregulated histone lactation modulate gene expression. So usually proteomic lab don't do genetics. we have a genetic department chair here, right? I never did genetic in animal model. We did knock out genes in cell line frequently, but animal model is too much for me. So we recently very lucky. We have a very talented postdoc who happened to be a cell biologist, has a lot of experience with animals. So we did conditional knockout. We do the cell type specific knockout of GTP-SCS G2 surveillance. And my next door neighbor, my collaborator, Lev Baker, he knocked out LDHA. So we knock out these two genes, not only in cancer cells, but also in macrophage. So from our two labs, our data clearly show that these two genes are in the same pathway. Both knockout can regulate macrophage's function and modulate macrophage M2 versus M1 ratio in tumor cells, and also can regulate tumor growth. So together, from this genetic data, we show that this pathway is physiologically relevant, at least in animal model, and it can regulate macrophage function, it can regulate tumor growth. So we hope we can cook out a story. This is a gigantic story from both labs. So in the close future. To summarize my talk, I just share with you L-lactate can be a precursor for lact-CoA. The lact-CoA is a substrate, is cofactor for histone lactation. And since we published our first paper about 5 years ago, and because lactase is so interesting and the people interested in tumor microenvironment hypoxia, so our paper has been cited for exactly 1,800 times as of today, actually. So we are very delighted. So histone L-lactation size are upregulated in response to hypoxia and tumor microenvironment. So L-lactate mediated or stimulated histone lactation pathway regulates gene expression, which is mechanistically different from the popular acylation and methylation pathways. So we detect 113 histone lactation sites, and we and many others carry out proteomic analysis with the advanced proteomic technology, and a typical proteomic study can identify up to 2,000 or even more lactation sites. We try to characterize biochemical regulation of this pathway. So we identify P300 and CBP and the MORPH as lactotransferases. Our community colleagues in the community identify HB1, GCM5 are also lactotransferases. We show that eraser HDAC 1, 2, 3 are the eraser proteins, and others show that some 33 family member can also catalyze histone delactation reactions. And some group also identify BRG1 as a reader specific for histone lactations. So a fundamental question will be for like a protein like P300, where is the specific? How the specificity is achieved between acylation and lactation. So there are a bunch of possibilities. Of course, you can argue specificity of lactotransferase, delactylation enzyme, and the reader proteins. So in our study, we show that compartmentalization is important. Also, the right, like a P300, and the enzyme for the lact-CoA synthesis, their complex or their interaction will be also important. In the case of P300 cannot catalyze lysine succinylation. So when these two complex together, so it's specific for lactation, not for acetylation or for succinylation. So we from diverse cell biology, genetics, and the clinical studies we carry out. So our result point out one direction. This pathway is physiologically relevant, and it can regulate macrophage function. It's important for tumor microenvironment. So in addition to lactation, we also identified 12 other new protein modifications. They are structurally very different from popular acylation and methylation. They can induce much more significant structure change than acylation and methylation. Therefore, functional is difficult to against their role in the biochemical regulation, at least from the protein chemistry point of view. And so a lot of studies were carried out in the past decade, including a recent study from Mike Snyder's lab, also Mike. So they show that propionylation is important. So indeed, propionylation is among the most abundant, I think, modification among these short chain modification. So in some cells, it can be more abundant than acylation. This is not from my lab's data, it's from some colleagues' data. So some of these modifications are associated with physiological change and genetic diseases. For example, for this genetic disease, historically, we only know the association between mutation of these proteins with this genetic disease. We don't know why, we don't know how. So identification of this new modification clearly suggests that these short-chain lipid and short-chain lipid modification are relevant. So as one example, like for example, in diabetic patient, beta-hydroxybutyrate concentration is stimulated for 10 to 50 fold, right? So beta-hydroxybutyrate can be the precursor for the synthesis of beta-hydroxybutyrate CoA, which in turn serve as a cofactor for histone beta-hydroxybutyrylation. So histone beta-hydroxybutyrylation can regulate gene expression. So in other genetic disease, for example, like short chain CoA synthesis deficiency, among those cells, propionylate and butyrate will be dramatically accumulated that lead to dysregulated histone butyrylation and propionylation. So as you know, these short chain lipid and short chain lipid CoA are important for the cellular function. Dysregulation of this molecule can lead to disease. For example, like melanin CoA is highly regulated. Concentration change of melanin CoA is associated with cardiovascular disease. So, these molecules are precursors or cofactors for these short chain lipid modifications. So under diverse cell conditions, genetic mutation, energy status, energy source, and the environment. So these short chain lipid or their CoAs or the activity of acyltransferase or deacylation can be changed. So leading to differential program of short chain lipid modifications. So this dysregulated short chain lipid modification can regulate cellular phenotype and the network. So I guess it's a complex problem. You can argue in some cases it's stemmed by phenomenon, in other cases it's important. So I hope the collective effort from the community will address these daunting challenges. Now you can argue these modification pathway create many problems, and here I just share some of them. For example, how these short chain lipid CoA are produced, how they are regulated. So at biochemistry level, what are the common and difference between this modification and the widely studied acylation and methylation? So what are their lead proteins? What are substrate proteins? What are the mechanism and the epigenetic regulations? So are there any synergistic effect or combinatorial effect between this modification and between this modification and acylation and methylation? So what are their contributions to diverse diseases? So as you know, microbiome produce a lot of short chain lipid, including propionate, butyrate, and others. So are these relevant? And in the tumor microenvironment, in addition to lactate, there are also other short chain lipid. So are these modifications involved? So what are their contributions to physiological changes and diseases? So hopefully we will have more answers in the future. Finally, I want to thank my colleagues in the lab who contribute these studies. Thank you again, Mike, for your invitation. So it's really very delightful to be here. Thank you for your attention. More than happy to entertain your questions.